Today I will be talking about things that I've been doing in my lab over the last 12 years or so since I moved to Harvard in 2010. And it is about what you see there, about making biochemistry free artificial life in a test tube, meaning trying to build life or systems that behave like natural living systems, except they do not have any biochemistry. It's a work which is totally out of the box, thinking out of the box, and you will see what comes out of it. So without any further ado, I will start talking, except that I would like to say that my plans is to talk about 45 minutes and then open the floor for discussion or questions or anything you want. And anything you want, any questions you may have. Okay. And so let me start. So why does a person like me, a physicist, begin to think about artificial life in a test tube? Why do you want to do that? In the 1990s, I became quite interested, whether with a former colleague of mine who died, who was a physicist, and shared with me the notion that the complexity that you see at the surface of things is a reflection of a very deep simplicity. We started thinking about a problem. We wanted to work together. We started thinking about a problem that would interest him and would interest me. We both are. He was very interested in birds, and so was I. So we started thinking about life. And then we became involved in the idea of pursuing this notion of working together. We became involved in something that's called astrobiology, trying to understand why life exists on Earth and why it may be in other places in the universe. So I started learning many things, and one of the things I learned quite quickly, or I should say, like Mark Twain would say, I was learned quite quickly, is that there is a lot of complexity already in the interstellar medium, chemical complexity in the interstellar medium. So it became something, thank you, it became something that we began to think, what would be the stock of chemicals that may have been on the Earth, early on the Earth, that may have remained here, that actually gave rise to life. At about that time also, after they corrected the myopy of the Hubble Space Telescope, the Hubble Space Telescope found, let me see if my pointer works. Can you see my pointer? The Hubble Space Telescope, once they fixed some major issues with the lenses in 1992, they discovered that if they turned the telescope around this area, or around this area, you would discover, if you looked in more detail, you will discover the presence of this large sized object, like 10,000 times, just to say a number, 10,000 times to 100,000 times the size of the solar system, which they call proplets, protoplanetary disks, and which were predicted by the Russians who wrote theories of the formation of planetary systems back in the 1950s, and they were predicted to exist in interstellar clouds. So you saw these systems in the pictures, and we began to think that maybe there will be many more planetary systems. And indeed, a few years later, 1995, began to discover the existence of many extrasolar planetary systems. So the notion of life become accepted and interesting to many people, including themselves. These pictures that you see here were made public last year on July 2023, and it's with a new version of the Space Telescope. They were taken with the James Webb Space Telescope. So we do now have many planetary systems. Today we have about 5,000 extrasolar planets in planetary systems. Most of them have, as we are able to see, have 2, 3, or 5 or so planets. So yourselves, what it is clear is that there is a huge variety of planetary systems. They are depicted here in some picture, and many of them are systems containing planets which are like ours, rocky planets. We don't know about the history of our planet. Well, if you want to know if life was started here, how it may have started, you need to consider the history of the planet. If you look and to consider the history of the planet, I will just put this slide here, which is a little bit antiquated, but the essence that it displays is still true. That the planet is about 4.6 billion years old. The way to remember the age of the planet is to say 4, 5, 6, 7. The age of the planet is actually 4.567 billion years. Okay, so that's easy to remember. Okay, and we do know that the planet started by accretion of a cloud, just like the one which is making the droplets, and today, which we see today, in line from here. And we do know that by about 13 billion years ago, we knew that the planet had stable hydrosphere. We do know that some chemistry happened between 4.2 and 3.8 billion years. Exactly what occurred here, this is called the photocell era. I also like to call it the generalized biology era. What do I call it, and what is generalized biology? A genius of science, whose name was John Desmond Burnham, in the 1960s, began mid-50s, 1960s, we went to think, everyone thinking about the notion of life, which is the same, it performs the same functions as natural life on Earth, but which may have different chemical compounds, may be based on a different chemistry, and so on. But the important thing is that it satisfies the set of requisites, just like you see in most life on planet Earth. So this is what I'd like to call the protocell or generalized biology era. And then we do think that came an era where the RNA molecule appeared and gave rise to what is called the RNA world, which is already extremely complicated. And we know that the first living systems, as we know them today, had a common ancestor, which is called Luca. In Spanish, Luca. I am from Spain originally. Luca, when somebody says that, you say the one with the wig. And it means Lucas el de la peluca, which means Luca, the one who wears a wig. So each time I see it, I don't think of the last universal common ancestor, but I think of Luca and his wig. So anyway, we do know that after Luca, until today, we do know that life is based on biochemistry. So what are the steps, or what steps may have taken place there? Well, people have not been very open to discuss that because the science was not developed well enough until the last 30 years or so. And today, people who work in the evolution of life itself, of course, we all know that current extended life on Earth is spread into three major domains: the archaea, the bacteria, and the eukaryota. But we know that the last universal common ancestor was about 3.8 or so billion years ago, give up plus minus 0.2 billion years, which is nothing compared with the 4.6 billion years of the planet. So in biology, like for example, my friend Andon, they begin to think about the necessity of having some further ages which had a different constitution than natural extant life today on Earth, and that eventually evolved into the most recent common ancestor of living things, Luca. So understanding what here is a major problem in origin of life. Little, if anything, is understood there. I think this community can contribute greatly for many people in origins of life to do or are not aware of many of the techniques that people in the artificial life community use. And so there you can find a lot of problems. And if you want to talk about, I mean, problems in the best sense of a science problem, a lot of science problems which we cannot very quickly, both problems which can be solved with computers, problems which can be addressed in the laboratory, and problems that can be addressed also by doing planetary system studies or observations. There are many space missions which are being prepared as we speak now to actually try and look for signs of life in extrasolar systems, in extrasolar systems and exoplanets. So there is a lot of opportunity for working these areas. And the work I'm going to be describing today has to do with this area, the area of the protocell or generalized life, as I like to call it. So what are the questions? Well, the question that we ask is all the above, all the properties all that characterizes living systems, living systems based on anything which is now current standard physical chemical processes? Of course not. Everything is based on standard physics and chemistry. And it is based, however, it is realized, at least since Luca, by using biochemistry. So there is a huge leap in complexity from chemistry itself to biochemistry. You look at textbooks and you see immediately what the great leap is on the two chemistries. And even from biochemistry, if you were to have biochemistry already available, there is also a great leap to extend life. So the question that I am asking and that I will be discussing in a couple of minutes as we go is, does the widely held and extensively validated principle of used in fundamental physics that the complexity of the surface is a reflection of a very deep simplicity? Think of the table of the elements, the periodic table of the elements. The nuclei, the nucleus there is made out of protons and neutrons. Protons and neutrons are made out of quarks. Simple. But when they get together, the whole is more than the simple sum of the parts. The word simple is very important there. 2 plus 2, 4. That's simple. 2 plus 2 more than 4 is not simple. Okay, it's much more complicated. Now many things come in. So to actually think about this and do something about this, we need to understand or know at least what is it that we mean by life. Of course, we all know that we look at life as we know, we see it today in our planet, natural life, extinct life. And we will see that there is a large variety of forms and so on and so forth, but they do things like they adapt, evolutionarily adapt. They manifest order, or they regulate their life, or they process energy to reproduce, self-reproduce, and so on and so forth. It turns out that, as discussed by many people, and you can, if you need the literature on this, I can help you find it. And by the way, at the end, I will put a transparency at the end with my address. If you have questions or comments or criticisms, well, you can send it to my address there. And I will also suggest a few references on reviews which are taking place, which have been written in the last few years. So what we do take as a characterization of life is chemical systems, which are actually capable of handling information. That is to say, they can generate information, they can transmit information, use it for storage, and so on and so forth, in order that the living system doesn't to invent the wheel over and over again. All living systems are capable of making their own parts and energy from simple parts that define energy, that define in the environment. They are capable of executing a form of self-reproduction, which is program, controlled self-reproduction, and they all experience or show adaptive and non-adaptive evolution, forms of adaptive or adaptive evolution. The question we want to ask is, can we integrate Can we unify? Is it possible to unify these properties, these functional properties, into making an aqueous artificial chemical system without using biochemistry? That's, as I said already, is what what I said will be the central topic. All living systems are actually unifying these properties in the so-called cell division cycle. The cell division cycle is a process that every living system obeys, and which can be summarized, or can be, summarized is a good word, to this graph here, which is important, and which is taken from a beautiful book on the cell division cycle written by Murray and Hunt, and published by Oxford University Press in 1993. Much to my surprise, in page 7 of the book, you find this graph. This graph tells you that every living system executes a process in which, if you represent the cell mass on the left and on the right, you represent the DNA per nucleus, and you sort of say that the amount of DNA per nucleus is coordinate to the amount of information that is contained in or is used by the system. So what you see, and this is of course an extreme idealization, is that the mass grows not linearly, but it grows from 1 unit to 2 units using metabolism, and then it reaches size, which is twice the original size in some way, and then the system reproduces, makes a second copy of it. At the same time that the mass is growing, the DNA at first stays more or less constant, and then there is a point where the DNA, the replication of the molecules of DNA, this is in extant systems today, the replication grows and grows to a value of 2, stays there, then it reaches a point where we identify it as chromosome segregation, and it grows, it makes 2 of that, and then eventually the mass has reached 3, then the system actually produces its reproduction. This is a unification of all the previous properties, and it's an integration of mass information and energy processing by life. Can you to do this? How would you do it? Well, as I said, I think in the first microsecond of the talk, I'm a physicist, so I think like a physicist. So I think in terms of equations, which describe properties, and those properties we try and unify them into one single form of equations, and then we test if those properties can be represented by the solutions to the equations. So the question is, can a generic mathematical physics and chemistry, high level, that is to say, little detail, top down, or outside in, as I prefer to use, because top down is confusing to many people, can we find an outside in representation of the properties of life that I showed to you earlier? And can you find solutions which actually attest to the fact that you are representing those properties? And then, if you are able to do this, can we use the above as a guide in the laboratory synthesis of some molecule, mimics of simple life, or maybe as originators of a simple life in terms of the notions that per now mentioned? Of course, in physics, we are very used to such notions. For example, Maxwell equations unify in a set of equations, right here in the middle, unify 2 very fundamental properties of matter: Coulomb's law, the electrostatic force, and the Savart law, the magnetic force. They both are described phenomenologically. They are described by this type of force, or in the case of the magnetic field, you have the description here. And these 2 were unified by the mathematical, physical, and so on and so forth genius of Maxwell when he wrote down his equations in 1962. All the properties of these are unified. When you use mathematics to solve these equations, you find out that straightforwardly, you find out that there is a prediction, the prediction of transverse waves, electromagnetic waves. And the waves, the velocity at which the waves propagate is also determined or predicted in terms of the permeability, so on, of the vacuum. It was until 1887, 25 years later, that it was discovered by Hertz that such waves actually exist, the electromagnetic waves. And then, of course, 10 years later, Marconi patented the radio, and here we are in the information technology era. So what would be necessary to write down a representation of the mathematical properties that we have said earlier a living system must have? Well, you dress yourself with the hat of the spherical cow, and you say, well, what is the minimum you will require? So you look at your hat and you find out that you want an environment, life exists in an environment, then you need food, and you also waste. Living systems are out of equilibrium and they produce waste in many ways. And then you also need the living system itself. So the simplest that you can think of, or at least that we could think of, and others before us, by the way, like Dante, his kingdom and so on, although this level of simplicity, you need to represent the chemistry, you need a mechanism to transport molecules, move molecules, another mechanism for molecules to actually react chemically, and this can be, for example, resolved by diffusion reaction of the molecules, as Trilling began to think about in the early 1950s. As I said, you need at least two types of substances. One, the cytoplasm, and another one to represent the food, and another one to represent waste, and an environment that you can model in principle by some random intrinsic component in the environment. So you can do that. You can represent those properties and then write down, constrain the equations that you get by using minimal mass action kinetics, and unifying and write down these equations. These equations are an extension of the Gray Scott model. Most people call it Gray Scott. Gray Scott started working on the model in the 1980s at best. Selkov had been working on it in Russia in the 1960s. Okay, and this that model contains two pieces which are very close to the first two equations there, reaction diffusion equations there, and then there is the third equation for the waste, which we added, constrained it by mass action kinetics, and we were very surprised to see that this principle leads to the Brusselator, except that there is a stochastic component for the environment in various of this. You can look at details of this for the two equations for the Selkov Gray Scott model in a paper in Physical Regulators 2003, and extended with renormalization calculations and so on in 2004. In the Physical Regulators, it shows that a conception such as what you have here about the two equations actually leads to very interesting solutions. More recently, my colleague from Galicia, Spain, and associated to my group in Harvard, and Dr. Perez and myself, we have a paper which appeared in Physics of Life Reviews in 2022, where we describe this in full detail. The notion of the living system is something with a membrane, more about that later, and it's permeable, and there exists a realization of each of these equations at each of the nodes in the system. When you put that in a computer and you solve it, and for certain choices of the parameters, the reaction constants and so on, not all reaction constants lead to this in the phase space, but the phase space is very well explored in that paper. But you get things like this. The red is food. And these steering patterns that you see there are actually generating systems which self-reproduce. Systems which they are the solutions to these equations, and they do this. You can look at it in a little bit more detail, and you can take one of them and do the computation. And when you do that, let's look at this. This is the food. This is the cytoplasm, and this is something which is a consequence of imposing discrete boundary conditions there. And here's what you get. And if you plot the mass and size of the living system, you get, for this guy in particular, you get how it grows in some arbitrary units. And you see the path it follows. If you now compute the information minus the entropy, actually, is what is computed here, you see that it follows a path like this. And you can see what it is doing, and so on. So, to make a long story short, if it can be made short, there is a lot of work that needs to be done on this, obviously. So you put here in parallel a parallel between the notion of the cell division cycle shown by actually a guy called Octen, and then taken up by Merrill Hunt in the book, and what you see in the, I hate to do this for the people who are online, but and you can see there is a parallel between the top part of the graph, which is the theory or the generic information that biologists handle, and this is what the system actually produces. So this is all very encouraging. And then if you are, as I am, from Andalusia, you always think of a great someone from Andalusia, in this case is Picasso, who used to say this sentence that you have here in blue: everything you can imagine is real. Well, that depends on how much wine you drink, how many joints you have smoked, and how happy or unhappy you are in a particular day, as we all know very well. Okay, the important thing is that in our case, we need to use the scientific method. That is to say, have a description in terms of first principles, and then realize that description in nature. But as you can read, if you're interested in this beautiful book, the person may even be here, I don't know. By bottom, the value is in the details. You can understand why I write all of this and this in here. It's not easy to see the details. To begin with, you need to understand that many people have thought about it before you about these things. An example in point is sitting there in the fifth line from the top, Kirsten Rasmussen, a Danish colleague, who began to think about many of these things and putting them together in the early 2000s. This is his transparency. I asked him for this transparency a few days ago, and he was nice enough to send it to me. So we began to think that there was a notion about a new physical chemical life that involves metabolism, information, and containment. This is before I started thinking about any of these things in reality. And this actually is something that came later, but it's great work, and I wanted to bring it to the attention of this community, which many of you probably know it, but just in case. Okay, so this type of thing is what is behind what is going to follow in the presentation. So I am putting a disclaimer that the first ones to do this. Many people have thought about this, including Ganti and others, and Sten, very much in particular, who actually took the troubles to make it using some form of extensions of biochemistry and so on. And I don't think they actually, oops, excuse me, that's on Valley, not me. I'm afraid of Zoom, you can imagine how much. So then how do you start the system? Well, if you look at the boundary conditions that were involved in the simulations as showed to you, you need to have something like a boundary. This is something which Sten was also saying, and many others have said before, but Sten in particular has said it. And you need a boundary because the chemical reactions that take place are very specific, and you cannot be in a dissolved environment. You need to have the system contained in such a way that you avoid what is called the geometric dimming, which is that if the system is not, if the concentrations are not high enough, then the chemical reactions will not go through. So the first thing you need is something like this. Then you need also to know what goes inside, what goes outside, and what is the membrane materials. Well, a few postdocs of mine and myself, we started thinking, how do we do this? And then we learned about something which we had known before that it existed. And that is something which is called PISA, polymerization-induced subassembly. Polymerization-induced subassembly is a chemical processing where you start off, it's a type of chemical reactions, a class of chemical reactions. This is extremely developed now, okay? Which you start with a homogeneous, with the hydrophilic, that is to say, or solubilic if you're not in water, but let's restrict ourselves to water. So you start with hydrophilic element, like for example, polyethylene glycol. You put attached to it some chain transfer agent. This is standard chemistry, standard polymer chemistry, with some materials which are actually able to find molecules which are pre-existing in the environment, in this case, water. And these molecules will be joined here by this polymerization process. And they will form an amphiphile, a molecule that contains a hydrophilic block, phobic block. The length of the hydrophilic block will stay fixed. The length of the hydrophobic block will grow. This is important. And you have there some of the, not necessarily first papers, the first papers are described in this paper here. But you do have some of the contributions. We discovered this, Jan Szymanski, one postdoc, so we discovered this without knowing that it had existed before. And we learned about it right away. The referees don't forgive you. So that's great. That's what the referee system is supposed to do, okay? What happens in such a system is this. As the polymerization takes place, you make the amphiphile. As you make the amphiphile in sufficient quantity, the amphiphile will get together and it will self-organize and then form first micelles, and as the length of the hydrophobic part grows, the morphology that is generated will change. And it goes from, the system goes from having the molecules assemble themselves as the long part of the hydrophobic part will grow, the molecules grow, they first make spheres, then we make what they call worms, which are actually cylindrical spheres, cylindrical cells, and then eventually they actually generate vesicles. And this is taken from a paper from 2016 by Cumming, Smith, and Arms, another physicist who was at Los Alamos as well in the mid 80s, in the mid 2000s, early 2000s. So the system can put together that when we use a particular type of polymerization reaction, then what happens is that these spheres of the cells are very uniform. The system doesn't really lead to anything easily and almost automatically to anything that is actually that can be handled in the laboratory. In the late 1990s, the Australians came up with a polymerization reaction, which is called reversible addition fragmentation chain transfer, which is capable of generating chain control, control of the length of the chain of the hydrophobic tails, for example, which is uniform, as opposed to traditional radical polymerization, where it is not uniform. But what that translates into is that you can actually apply raft to PISA, And general systems, which are very those systems, for example, are described in this paper with another one of my postdocs, Juan Ren, a person from China. She is a very bright chemist. It leads to objects of this type. The sizes here are typically of the order of 20 microns. And the reasons why that's the size is because we chose the conditions of the chemistry in such a way that we could see it in a microscope. Why? Because we wanted to test what did these things do in the microscope. The reactions were taking place. We wanted to see it. Otherwise, it's mental elucubrations, so to speak. So we wanted to test this experimentally. So all that you will see that for me, it's all experimental work. And we have a setup to do that. It's a simple setup. You have it there. It consists, everything that is here is less than $2,000. The optical board is more expensive than the equipment that you have there, okay? So you have a pump, a rotary pump. It's called Wenger pump. Wenger is a person's name to actually cool a reactor here, which is a keyboard. Out. At first, we used fancy LEDs and all that. We use Christmas tree LEDs, which is great. And it actually works pretty well. And when spectrometers and this is a spectrophotometer of today. This object, this little box there, which looks like a pack of cigarettes. For those who don't smoke and have never seen a pack of cigarettes, it's about the size. It's smaller than my fist, and that's a very fancy spectrophotometer. So then we do the polarization during 16 hours in the thermostat in the barrel, in the outbound. We put all of this under darkness, and we mix it, and we put it under darkness because we need to show that it is the chemistry that actually does what it does. So the darkness is a shoebox over the black tape inside to make sure that it is dark. Okay, and the component, and this is very surprising, is that the chemical substances that you put into it is water, some monomer that you buy, like acrylonitrile or HPMA, that you buy from a chemical supply company like Sigma-Aldrich, polyethylene glycol, a raft agent, which contains sulfurs, which actually generate the appropriate conditions for the polymerization reaction with thinning to actually which absorbs the light in such a way that it accelerates the reaction. And then in this particular type of experiments, you take it, you start the experiment, you see how this evolves, you take a sample every over hour or half an hour, you stain it with fluorescent dye and observe it under a microscope. It continues evolution, and oxygen starved. Why oxygen starved? Oxygen is the enemy of polymerization. Okay, it poisons, it makes something which is called a singlet, and the oxygen singlet kills the polymerization reaction. So you start with, you expect the oxygen in the atmosphere that you do by blowing nitrogen. Today, the system is much more complicated, even more. So this is 3 centimeters in diameter. And we have this here. All the wall is covered with Christmas tree LEDs. And the system is put, this is a vial here. And this is what you can see happens in the laboratory with the microscopes. Today we have a very sophisticated size microscope, completely not completely automated, but completely instrumented to do all this. And we have completely autofocus lab microscopes, which are a marvel to work with, like this EVOS microscope from Thermo Scientific. And we, of course, do everything in darkness by putting shoeboxes essentially on top of it. At the door of my laboratory, there's this sign to make sure that nobody contaminates anything or anything. No biological materials allowed except for researchers, of course. So what happens? Well, you start the reaction, and when this is happening, 2 hours, you start, it's completely homogeneous. And the reaction of the mixture of chemicals I just mentioned to you, the reaction starts dark and transparent, homogeneous. It's also stirred at about 600 RPM. Then the system begins to react under the influence of the light. And eventually, after 6 hours, the system becomes opalescent. That means that there is phase separation, and that means that the processes of PISA have started. So you will get something here that you need to study. And here is what you do find there. So in this phase separated material, first, the full news. You get, this is what I just showed to you. These are a couple of pictures of what happens. This is a show to you in a minute. And what you do see is that out of the optical microscope starts kicking in, which is for sizes of a micron cell region, is that the molecules that we, the alpha files that have formed and that we have been formed because we do resonance, we do all the protocols necessary to understand the molecules. We take, so we do that the cell assembled into first made cells with electron transmission electron microscopy, scanning electron microscopy, and so on. We do see those molecules making cells. See this view, see from the first, we see somebody turning. And that is already a vesicle that has been formed by the evolution of the cells into a vesicle. Let me show you this movie. I'll show you this movie again later. This is important that you see it now. So you see how these guys are tumbling. The maximum intensity of the light is in the center. And you see how these guys are tumbling. Let's pay attention to this fellow here. It will keep growing. And the membrane is like a liquid that is expanding as it is growing. So there's a force there. Evolution, the chemical evolution of the membrane is described by a form of the Navier-Stokes equations describing the fluid membrane, which is called the Rayleigh-Set equation. Okay, and it describes the modes of evolution. What you find that this guy has found this membrane is tumbling. It's way there. It's actually some sort of chemotaxis. Its size represented here, and it grows. Well, then it collapses. You see that's when it collapses. It grows, collapses, and grows, collapses in both directions of the cell division cycle. So here we go. And in the interest of time, I just let it to, and you see that there are many more that are found, that appear. When this guy, for example, has collapsed and he has started to grow again in one of his cycles. We call this phoenix. You can count the number of vesicles in the system. It follows a path like this. You can check and see what is good. You can see the parallel between what the cell division cycle does and what these guys are doing, the chemistry which is taking place. We got this guys number in the system using some form of effect. We do know that the evaluation of the chemicals plays a very important role. And we do know that this process happens this type of reactions. You can also connect it with other complex reactions. the raft, see if it's connected with all reactions, like the values of subtle scale reaction right there. You can actually make some of these guys a population, mixed populations of these guys, and you can mix two populations, one that contains a catalyst and which does not contain the catalyst. So this is the one that contains the catalyst. These are the ones which don't contain the catalyst. They will share the food. And if you, in this case, if you share green light in these examples, and food, then of course, for the catalyst, are able to move faster, the cell moves faster, the ones without the catalyst, the cell reproduce also, but at a slower rate. And when you have a lot of food, then almost nothing happens. And then you can actually track what is going on in two populations using fluorescence microscopy. And you find something amazing, which is the way skies, the ones with without simple populations. Well, it was found by Rouse, his famous work on the struggle for existence for Alicia. So there is so basically a parallel between the way the skies evolve and actual process we see in life in the interest of time and just grow and you wrap up in some way. We start with some equations. We are motivated by how all life on Earth shares a series of properties. We write some equations for that. To actually realize those equations, we make different types of systems, not as complicated as this, which is a natural system, but something which is much more simple and much more idealized in some sense. And we have, like, if you are aficionado to coffee, we have like the Chemex version. It's of life. And this is a machine, a machine like the one in the room I am in the hotel here. Okay, who knows how the hell it works, but it does work. And so this is what we have. And so what it is true is that we are at the beginnings of a new era when people are beginning to observe nature. See, for example, the case of birds flying and all that, and we've been inspired by this. And in Rosalind found his equations, and then our friend Lilienthal and others learned how to incorporate this into wheels, and then unfortunately Lilienthal powered flight, so he crashed and crashed his skull. And then, of course, the Wright brothers, sometimes I think it is without the W, the Wright brothers took his work and then they actually made a power flight. Today, we are able to, for our systems, we are able to actually extend what I have shown to you to systems with more than one catalyst. See that the systems adapt. You begin to see that there is something, the door is open to what Stephen Jay Gould wrote after the Martian Uterite, which is that the origin of life. He wrote this as an editorial in August 11th of 1996. The origin of life may be virtually a consequence of carbon chemistry and the physics of self-organizing systems, given favorable balance and requisite inorganic constituents. So that's where we are. We do have several conclusions, but I'm not going to show them, except that they say that there is a lot of opportunity for experiment. There is a lot of opportunity for thinking, for doing theory, for doing chemical engineering around this. We are supported by these wonderful sessions. And if you're interested in this, just get in touch with me, and so you can take. That's easier to find than what is coming in this. And these are some steps that you can take a look at. like, for example, in the case of filming, somebody who just graduated, and it appeared on July the 5th. So these are selected effort uh, reviews that you can actually use to learn this stuff. So where I will start, I know that I've spoken about 40 minutes, 50 minutes, and if the organizer lets some questions, it will be great for me. So thank you very much. Thank you. Thank you. Thank you. Thank you.
Great talk, very inspiring. So maybe we get some questions from the audience. Yeah.
Thank you.
Hello, can you hear me? Yeah. Thank you, Juan, for the presentation. I have a technical question because I worked for years on replicating vesicles. In your system, when you have these self-replicating polymerized systems of vesicles, there's a thresholding effect where this large vesicle then pops, right?
Yep.
What's the basis? What's the technical physical basis for that popping?
The popping is predicted by the Rayleigh-Preset equations, which tell you that the surface energy is connected with the rate at which the surface energy of the vesicle is connected with the rate at which the vesicle expands. And it's also connected with the osmotic pressure difference between the inside and the outside. So what happens is that the system begins to grow, the polarization reaction begins to grow, the inside of the vesicles organize, and when they organize, they organize the molecules. This is a work in progress. Okay, so I don't know if it will work. So the are incorporated, and then they keep growing, then they self-assemble into many cells, and there is chemistry going on here inside. Was captured. Okay, that makes the vesicles grow.
Right.
And it's a force, a chemical force. So there is an acceleration. That acceleration keeps the system going. Then there is inverted micelles that from the inside. There is wear forms that comes in, and then there are defects. And the defects actually generate, weaken the surface energy. And that is something which leads to cavitation. This is one example. There are other pathways which do not involve cavitation. When the cavitation takes place, or when the breaking takes place, the systems actually emit what is inside of the reactive molecules which are inside. And that actually becomes the seed for children.
Right. So just so I understand I understand the growth process, which you have two different kinetic processes. The growth is slow, and this division is fast.
Yes.
The division is fast because it's a hydrodynamically controlled process.
Yes.
So it's like a bursting bubble.
That's exactly bursting, except that it is collapsing.
And it collapses.
Like in your car, when you try to accelerate and you have too much air, or I forgot if it is too much air or too much gas. I forgot what it is, but it makes bubbles and the car knocks, clock, 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 clock. So that is exactly the same process.
Okay.
It's extremely well understood. It's called cavitation.
Okay. Thank you.
Thank you. There's another question there.
Hi, thank you for a really lovely talk. I was really interested in your views. You spoke a lot about this, setting the system up and then seeing what it does. And when you were talking about the biology, you also spoke about kind of information and evolution. So in these systems, how do you embed that kind of aspect, the information aspect?
Yes,
in terms
the information. Yes, the information here is conformational information, which is the information contained in the molecules so that they react. This is not polymeric information like what you have in DNA. It's much more primitive. and anything like that, and information in the form of a polymer. Okay, information is in how the molecules, the geometry of the molecules, how they actually conform together, so that they actually give rise to this particular type of systems. Okay, and the systems, as information grow, and they're based on that information. So the information is put in there to begin with by you. That is to say, the environment, the emotion behind this, the ideology, if you wish, behind this, is that the environment provides you with all the necessary ingredients. Okay, and then it's called a heterotrophic origin. Then the systems begin to evolve. How do they evolve? Because that's goes together with your question. The systems evolve because a lot of junk comes out of them from the collapse, for example. The junk is not really junk, it's partially reacted amplifiers, which form self-ingredients which they can keep growing because there is enough food. And then these guys don't all have the same size. So but they are the basis. They have the basic information to actually make, in that particular environment, more children. But the children are not the same. There's heritable variation in the system, which as people evolution are aware of, that's a key basis for the winning evolution. Okay, so that happens here. And this competition, which occurs, also is fundamental in order to have evolution. Thank you.
Thank you again for the talk. Really loved it. On this topic, there seems to be, though, like a discrepancy here between the collapse of a vesicle and the emission of particles for replication versus what you see in cells of division, specifically taking one membrane and turning it into two independent membranes. Is there work you guys have done to analyze specifically how you create two membranes as a consequence of one?
Yeah. What I'm showing you is this expansion and collapse is because it's the one that we have had for a longer time, okay? And the one where we thought a lot about, and the one that inspired us to think. So I had a choice when I prepared my slides, and the choice to present what. And I chose this because it puts together the physics of cavitation, the growth of the values, and so on. There are other modes of growth, and there are other modes of reproduction. We have examples in order to fully understand with this detailed description. We have examples of actually division, okay? And the examples which are actually division, I can show you a microscope slide of that. And things like this. So this is not an optical microscope image, okay? These are dead but cut in the act. And this is how the optical breaks. And then you can make your numbers from there. Although it's very difficult to get real precise measurements on this, but it can be done. Giving enough money, enough talented people like this community, or a part of this community anyway, you can actually get a lot of data and check. What is important about this is that it is the simplest you can think of, or at least we can think of, and confirms to principles that people have thought for a long time, like the thing I mentioned about Sten Rasmussen. So people have been thinking about this, and this is a chemical, very simple realization of what is contained in these systems. So the answer is yes, we have examples, but we would like to have many more. We also, in the West, where we have the competition, there is also another way of reproduction there, which is the one that I showed to you with the phoenix is by squirting inside. There is another option which is squeezing by means of osmotic forces, squeezing molecules in the membrane and then exposing them from the membrane. And that we have completely documented. It's in a paper which appeared in Cell Reports Physical Science in 2023, if you're interested. Thank you for your question.
Thank you.
There is a person there in this fifth row. Oh, there's a person there also.
Yeah, thanks for the talk. I have a question regarding the first part of the talk about the equations of life, the 3 or 4 equations.
Hang on a second. Let me get close to you because
Yeah.
as you get older, you begin to get leaks. One of mine is that I'm losing hearing. COVID left me a beautiful gift, which is called diabetes type 2. And
Sorry, my voice is also low. So thanks for the talk again. And my question is about the first part of the talk with the equations of life, the 3 to 4 equations.
Yeah,
that
3.
you showed. Yeah, okay, 3. So I noted that the environment is uniform in food, like the distribution is uniform, but I think it wasn't like that because if it was the case, we wouldn't be here today, I think.
Yeah.
So is it possible, or did you think of amending the equations to take into account non-uniform environments and that the cells can like evolve to store this information?
Yes, I understand your question, and I will show you a component in the equations that I highlighted. It's a very good question, by the way, like all the questions. But I will show you how we account for that. So we added in the equations that I wrote down, which are written down here in a somewhat masochistic way. This is the diffusion part. Okay, this is an autocatalytic term. This is a feed term. This is another feed term of some kind. And then there are here these stochastic components. Those stochastic components are representing, for example, changes in the flow of E or V or amounts in the waste. They are stochastic. They are time dependent, but also space dependent. They're power laws. Okay, power law is here. You can explore to your heart's contempt types of noise that you put there, and they tend to lead to behaviors like the one you saw. So we have explored that. Much more work, of course, needs to be done. But what we have explored supports the notion that the learning can be represented by a stochastic term. Okay, thank you so much. Thank you.